Okay, so let's get started. Um, we covered biosignaling, membrane dynamics portions already. So we'll just go straight to chapter 12, which is on the biosignaling, right? And so signaling is very, very important features of um, all living systems. And for example, cells from one part of your body actually are in contact with other cells that might be really far apart, right? And how are they in contact? Um, Cells actually have developed mechanisms by which they can release certain signaling molecules that can, let's say, travel through the blood, if it's an hormone, for example, and go to a totally distant part of, of another type of cells in your body, and then go and bind to that, bind to certain receptors on those cells and elicit a physiological response, right? Cell signaling is really, really absolutely, as I always say, fascinating um, area of biochemistry. And a lot of times people are spending their entire life studying a signaling pathway. Uh, so there are four key features of signal transduction systems, and that's what is shown here in this slide. It turns out that whatever that signal is, as I said, signal could be, a small molecule, or it could be a hormone, or it could be another protein, and so on. Okay, and these signals are actually disposable chemical entities. When they go and bind to their receptor, they basically elicit a physiological response, okay, whatever that response be. So for example, insulin might bind. Insulin is a hormone released from beta pancreatic cells. It may be released from pancreatic cells, goes to its receptor, let's say a muscle cell receptor. When it binds, the net effect of that binding of signal to the receptor would be to bring glucose molecule in for metabolism, for generating ATP, for example. Okay, so one of the key features of the signal transduction system is that signal molecules are very specific to their receptors. Okay, so they only bind to their receptors. If there are other receptors, they're not going to affect the other receptors. They won't go and bind. And once they bind, that signal that you are seeing here, S1, for example, that signal gets amplified that's the second feature of the signal transduction network. Okay, and typically what happens is that signal will go and bind to first enzyme in the cascade. And that enzyme will actually create its own signal. So you can see that enzyme 1 is able to actually create three signals, right? Three molecules here as shown here in the green triangle in a circle. And those will go and bind to, let's say, three molecules of enzyme 2. And then each of those enzyme 2 molecules will go and release another three molecules, signaling molecules, and they will go on to bind to enzyme 3 and so on. So this is what we call signaling cascades. You can see that when enzyme activates another enzyme, in this case enzyme 1 activating enzyme 2, enzyme 2 activating enzyme 3, the number of effective molecules increases in a geometric fashion. It's not linear. It's not like 1, 2, 4, 6. It's 1, 3, 9. So that's how signal gets quickly amplified. Okay. And sometimes once the signal, that pathway needs to be turned off, for example, let's say signal was to bring glucose molecule in, but all glucose molecules already got into the cell. Now the blood glucose level is a standard 5 millimolar. Then that signal, then that signaling molecule actually leaves the receptor, and receptor gets desensitized. Okay. So many times when you do not need, when the signal has fallen off, receptor will be taken up by the cell, and it will get internalized and then maybe degraded into its constituent components. Okay. The other thing is that sometimes response itself can actually act as a feedback circuit. Okay. So if you are generating a response, which could be another signaling molecule, and that signaling molecule is not being utilized in the next cascade, it accumulates, then that response, that molecule will go and bind to the receptor and inhibit its activity. Okay, so that's what desensitization and adaptation means. The fourth and final one is integration. Okay, so this is a very interesting one that you should pay attention to, and I will show you one signaling cascades where you will see how two different signals can be integrated to create one individual response at downstream of the signaling pathways. Okay, so you can see here that signal one, when it's binding to receptor one, the net output is that you are increasing the concentration of X, or you are actually increasing the voltage, Vm. Okay. On the other hand, when signal 2 binds to the receptor 2, its own receptor, net output of that signaling cascade is that it decreases both the concentration of X and Vm. Okay. So when both signal 1 and signal 2 molecule binds to their respective receptors, the net outcome is just the algebraic sum of the two outputs. Okay, so let's say when the signal 1 bound to receptor 1, 10 molecules of X produced, right? And when signal 2 bounds to receptor 2, 5 molecules of X went down. Okay, so whatever the concentration was. So the net will be 10 minus 5. That's what the net delta X will be. And that's the integrated response. Okay, so when two signals have opposite effects on a metabolic characteristic, such as the concentration of, let's say, second messenger X, or membrane potential in this case, the regulatory outcome overall results from the integrated input from the both receptors. That's what it means. Okay, so signals can integrate downstream. Okay, so that's what basics of signal transduction is. Now, some signals to which cells respond are shown here, and many of them you will see in the coming lectures when we talk about metabolism, right? You will see certain developmental signals, growth factors, hormones, neurotransmitters. When they bind to their receptors, their cells respond. Okay. So in terms of types of signal transducers, there are 6 different types. Okay, 6 general types, and those are the ones that are shown here. Okay, so in the extreme left, you are seeing these G protein coupled receptors, right? You can see that there is a receptor here embedded in the plasma membrane, right? And when signal binds to the outside of the extracellular domain of the receptor, what will happen is that there is a receptor here that gets activated. Okay, and that activation of receptor upon binding of the signaling molecule leads to the activation of what here we call it G protein. Okay, G protein is a specialized protein that gets activated when GTP is bound to it. Okay, and it exists in the inactive state when GDP is bound to it. Okay, GDP GTP exchange, that's what turns this G proteins on and off. Okay, and there are variety of G proteins. So it will activate a G protein, intracellular G protein. You can see that it is on the cytoplasmic side, right? Signal is binding outside, but G protein gets activated in the cytoplasm, right? And so then once G protein is activated, it can regulate the activity of another enzyme that could actually generate a second messenger like X. Okay, and that's how signal is passed on to this type of system, in this type of system. Then you have receptor tyrosine kinase, and I will show you particularly, this is the reason why I have boxed this one, because we will talk about insulin receptor signaling in a minute. So these are receptor tyrosine kinases where they typically exist in the dimeric form, okay, and they have extracellular domains where the ligand can bind, signal can bind. Okay, so here they're showing you that this is a dimeric protein where S goes and binds to each individual units on the extracellular matrix side. And when the signal binds, it actually activates a tyrosine kinase activity in the cytoplasmic side. Okay, so that's what they're showing you in the blue here in the cytoplasmic side. You can see that they are showing you all these lines coming from it, basically trying to tell you that this receptor is now activated in the cytoplasmic side, and that activation is of a tyrosine kinase activity. What does that mean? That usually means that it will have a kinase activity where it will take gamma phosphate from ATP and transfer it to a tyrosine residue. That's what tyrosine kinase activity means. And the way tyrosine kinase activity gets built in here when the signal binds is that these two receptors, they literally transphosphorylate each other. Okay, so one unit will go and transphosphorylates, let's say 3 tyrosine residue on the other one, and the other one will go and transphosphorylate 3 tyrosine residues on the other one. Okay, mutual. That's why it's called autophosphorylation. They literally autophosphorylate. And so then what happens once the kinase that is active, it will go and phosphorylate some other substrates downstream. And eventually what will happen is that signal will go to the nucleus. It could be in the form of transcription factor. Transcription factor will get activated. You will produce messenger RNA for that gene, and then ultimately will produce the protein. Okay. So that's the second type, and we'll discuss that a little bit in length in this class. The receptor guanylyl cyclase is when, it's typically a monomeric protein. When it binds to a signal, the cytoplasmic side of that protein will get activated. And when it gets activated, it will actually take GTP molecule, not ATP, but GTP molecule, and convert that to cyclic GMP molecule. Okay, cyclic GMP is a second messenger that will go and bind and activate some other proteins. And that's how this signaling happens. Fourth one is this gated ion channels. If you recall, we talked about how a ligand can bind and open up the channel, right? So here you can see that signal binds to the site to the extracellular phase of the receptor, and once it binds, it actually opens up the channel, right? And then whatever ions has to go can go in, and so on. The fifth type is signal which doesn't require any type of interactions at the membrane bilayer. Basically, it's usually a hydrophobic molecule like a steroid that will enter the cell, cross the plasma membrane without any problem, just by being very hydrophobic. It will be able to cross the membrane on its own. And then it can go and bind to certain nuclear receptors that are found in the nucleus, and then allow that nuclear receptor to regulate the expression of specific genes. Okay, so that's the fifth kind. And the sixth type is the adhesion receptor. An example will be integrin. We talked about integrin a little bit in the last lectures. And so here what you do is you have, they're showing you, this proteoglycan structures, right? And typically they also act as a dimer, okay? And they, when something binds, molecules bind in the extracellular matrix, it will change the conformation on the cytoplasmic side, okay? And then that conformational change will be conveyed by its interaction with certain other proteins, cytoplasmic proteins, could be, involved in rearrangements of cytoskeleton, for example. Okay, so yeah, these are the 6 general type of signal transducers that you should know at the minimum. So can we talk about the specific? We're not doing that many slides on signaling pathways, but can we learn something? Maybe one specific or 2 specific pathways, so we can get a feel for how this thing happens. Okay, so here is, if you pay attention, I'm showing you in A an insulin receptor to which insulin is bound. Okay, so if you look here carefully, you'll see that there are 2 transmembrane domain beta ones, right? And then you have 2 alpha domains that sits outside the membrane. And literally between those 2 alpha and 2 beta domains is where the insulin molecules binds. Okay, so you can see one insulin molecule binds to each one of those receptor on the outside. And it's, how do we know that? Because crystal structures are known. If you see here code, PDB database code, you type in PDB ID 2CEU in Google, and it will pull up the structure for you, and you can actually look at the structure, rotate the protein, and see how insulin is bound into the receptors. Okay. But and I think if I remember, still the structure of the transmembrane domain is not yet solved. Okay, but we know a lot about these domains that exist on the cytoplasmic side. The blue portion shown here in A, the bottom one. And so we know when insulin binds to insulin receptor, these 2 beta domains on the cytoplasmic phase will go and transphosphorylate each other. Okay, so in C they are trying to show you what residues get transphosphorylated. Okay, and what are those residues? So in C on the left hand side, what you're seeing is an insulin receptor, the cytoplasmic side of it, when it is in the inactive state. How do you make the crystal structure of that? That's very simple, right? You don't add any insulin, and in that case, if you have insulin receptor, it will exist in the inactive state. So in the inactive state, which is when it's not autophosphorylated, you'll notice that there are 4 residues that play a critical role. Okay, so there is tyrosine 1158, tyrosine 1162, and they're showing you nicely here in the black color and the red color. Red color is the oxygen of that tyrosine. Okay, and then tyrosine 1163. So those are the 3 tyrosine residues that exist like that in the inactive state of insulin receptor. And particularly this tyrosine 1162, right? It's hydrogen bonded to an aspartate residue, aspartate 1132. And that hydrogen bonding keeps this activation loop shown here in the blue, okay, in this place. This hydrogen bonding network, tyrosine 1162 to aspartate 1132, that hydrogen bonding network stabilizes this activation loop in such a way that it literally blocks the binding site for the substrate. Remember, what happens is this insulin receptor gets activated and it has to go and phosphorylate its downstream signaling molecules. That's what its function is. So the downstream protein molecules must have access to this kinase domain. But in the inactive state, that thing is blocked. But once insulin goes and binds, and the beta domains, they transphosphorylate each other on 3 tyrosine residues shown here, then amazing things happen. Okay, one thing that happens is that this whole loop that you are seeing in the blue, that actually swings out by almost 30 angstrom, okay, and creates a binding site for the target protein to come and get phosphorylated. Okay, and so why does it loop swing so much, 30 angstrom? Basically what happens is it's all about chemistry. Once these tyrosine residues get phosphorylated, those phosphate groups have large amount of negative charges on each of them, right? And so though they repel each other and they try to swing away from each other so that they can minimize the electrostatic repulsion. And you can see that how tyrosines in the active state are all facing away from each other, including aspartate 1132, right? Beautiful. How cool this is. Think about it. This really happens. Okay. So can I show you on a broader scale, on a global scale, how the signaling happens when it comes to insulin receptor pathways? That's what is shown here. Okay, and 2 things I want to show you in this slide, and I want you to take home. One is the individual interactions play a critical role in signaling. Okay, and you will see that how. The other thing I want to do is that I want you to see how sometimes certain types of signaling networks are integrated together. Okay, and I'm talking about here in B 2 different pathways. One is insulin, and the other one is leptin. Leptin is also a hormone. Okay, leptin is a hormone that is released in your blood, and that's what tells your brain, hypothalamus, that when you are eating and you feel full, you feel the feelings of satiation. That's because the leptin has been released by your adipocytes, and that leptin is actually going and binding to the leptin receptor in your brain and telling you to stop eating. Okay, that's so in some people who actually are very prone to obesity, this leptin receptor pathways are messed up. It's not working efficiently. And we will talk about more about that a little bit later on when we talk about metabolism. Okay, but in A, what I'm showing you is that certain people have propensity, like susceptibility to towards obesity. Okay, they eat little, but they tend to put on weight. And if they don't take control of their obesity, eventually what will happen is that obesity will lead to type 2 diabetes, okay, which will be insulin resistance. Once you have insulin resistance, you will have impaired glucose tolerance. Okay, the amount of glucose in your blood cannot be controlled properly, and that will lead to a lot of problems. You will develop type 2 diabetes, and if you don't manage your type 2 diabetes, you will end up actually destroying your beta cells of the pancreas, which produces insulin and releases insulin. And then if you don't even take care of that, then lots of trouble. You know, you'll lose your eyesight, you'll have your kidneys dysfunctioning, you know, tons of problems. Okay, so you can see that looks like obesity is linked to sugar metabolism. insulin signaling pathway, right? How are they linked? And I'm showing you at the bottom a very interesting signaling slide that actually took from Nature Drug Discovery Journal. Okay, so here is what happens once you eat food, carb-rich food. Insulin is released into your bloodstream. It will go and bind to insulin receptors, let's say in your skeletal muscles, and insulin receptor will autophosphorylate each other, remember, and 3 tyrosine residues. That's what they are showing you here, right here, on the cytoplasmic side. Once the tyrosine kinase domain of insulin receptor is activated, it will go and phosphorylate another substrate called insulin receptor substrate. Okay, there are 3 different types, 1, 2, 3. So insulin receptor substrate will get phosphorylated. Once insulin receptor substrate gets phosphorylated, it will go and bind and activate PI3 kinase. It's a kinase. Okay. PI3 kinase, what does it do? It basically will take phosphatidyl inositol for 4,5 bisphosphate and basically will convert it to inositol 3,4,5 triphosphate. Okay, that's what the chemistry does. But anyway, it's basically will generate phosphatidyl inositol 3,4,5 triphosphate, as you are seeing here, right? This molecule will go and bind to a kinase called PDK1. And PDK, once it's bound to PDK1, PDK1 will go and activate a very, very important protein called PKB, protein kinase B, are also known as AKT. Okay, AKT and PKB are the same. You will see different names, different places. But eventually, once AKT or PKB is activated, the net effect of this insulin binding to insulin receptor is that glucose molecules will be brought in. Okay, there is a transporter called GLUT4. GLUT4 will translocate itself to the membrane and will start bringing glucose molecule in for metabolism. Okay, so that's what happens. So this is how glucose is brought into the cell. Cool. Now, it turns out a protein enzyme that I have worked actually for 5 years almost on this enzyme called PTP1B. Okay, PTP1B can be localized in the cytoplasm, or it can also be localized into the ER, endoplasmic reticulum. And what PTP1B does is that it is a negative regulator of insulin receptor signaling pathway. Okay, what do I mean? What do I mean by that? It basically will negatively regulate this pathway. Okay, so it will stop the pathway. How does it do? When glucose levels are normalized in your blood, insulin concentration goes low, back to normal or low in your blood, then this signal must be turned off. And that signaling is turned off by PTP1B. It will go and dephosphorylate insulin receptor substrates. It will go and dephosphorylate insulin receptor itself. It's a phosphatase. And thereby it will cut off the signal. Okay. So myself and many others have actually tried to synthesize inhibitors of PTP1B to treat type 2 diabetes. Now I will let you think about that. Why inhibition of PTP1B would be good for a diabetic patient, especially type 2 diabetes, the people who have developed insulin resistance. I don't want to leave you in limbo because I know many of you will not go back and think about it. So let me tell you why. Because if you inhibit the function of PTP1B with, let's say, a competitive inhibitor, what will happen? This insulin receptor signaling pathway will go on and on, even when the glucose level has, even when the insulin has been removed. Because there is no PTP1B to dephosphorylate insulin receptor and turn off the signaling. So therefore, in those patients, you will actually continue to bring glucose molecule in and metabolize, and therefore you'll try to normalize the sugar level in the blood. I will talk about more about that a little bit later on. Okay, but I just gave you away the answer. So this is how scientists think about things, right? This is how you discover drug. Now let me quickly tell you leptin. Leptin is the hormone that binds to the obesity receptor. And when it binds to the obesity receptor, there is a kinase called janus activated kinase, JAK2. JAK2 gets phosphorylated. Okay. Once JAK2 gets phosphorylated, it will actually activate its partner STAT3. Okay. Also by phosphorylation. STAT3, once it is activated, it will actually go to the nucleus and it will reduce fatty acid synthesis, okay, and try to increase the fatty acid oxidation pathway, okay. These pathways of synthesis and oxidations of fatty acids are reciprocally regulated, which means it makes sense when you are trying to synthesize fatty acid, you don't want to burn fatty acid at the same time, right? And so some of these pathways of synthesis and breakdowns are reciprocally regulated. When one is active, the other one is off. Anyway, so this is one way you can see, but cool thing, look at here, integration. It turns out that PTP1B also dephosphorylates JAK2, okay? And so this is one molecule that actually can regulate both insulin receptor pathway and leptin receptor pathways, right? And so PTP1B inhibitors are also known to actually known to be as a potential drug for anti-obesity. Cool, right? Now I have to show you a pathway that you can appreciate a little more, okay? And that's what I'm going to show you, a MAP kinase pathway. MAP kinase pathway is very important when the cells have to undergo mitosis, okay? When cells have to proliferate. And so insulin, it turns out, is also a signal, positive signal for MAP kinase pathway. How does that happen? Insulin not only actually, activates the pathway that leads to the metabolism of glucose, but it also regulates pathways that are involved in cell proliferation. So that's what I'm trying to show you here in this very, very busy slide. Again, the same thing, insulin receptor will bind insulin, right? And then on the cytoplasmic side, the receptor will go autophosphorylations on 3 tyrosine residues. That's what is shown here. And then insulin receptor substrate 1 will go and gets phosphorylated on 3 residues as well by on the tyrosine by insulin receptor. So they are showing you IRS1 with 3 phosphate groups on it. Okay. Once you form this insulin receptor IRS1 triphosphorylated, the next protein in the cascade is this GRUB2. Okay, GRUB2 is a protein that has an SH2 domain. What are SH2 domain? SH2 domains are these domains in the protein that will bind phosphorylated tyrosine. Cool. So GRUB2 SH2 domain will go and dock itself on to the IRS1. And once it does that, beautifully, it opens up a binding pocket for this protein called son of 7-less, SOS. Okay, SOS will go and bind. SOS once binds to GRUB2, that's a signal for RAS to become activated. Okay, so RAS, what it will do, it will exchange, remember in lipid raft, prenulated RAS is often found outside of the lipid raft. Yeah, that same RAS. RAS will get activated by exchanging GDP for GTP. Okay. Once RAS is activated, activated RAS will go and bind to RAF1, again, a kinase. Okay. RAF1 will go and bind to MEK, and it will go and phosphorylated MEK on two serine residues. Okay, so RAF is a serine threonine kinase. Okay, and that will go and phosphorylate MEK on 2 serine residues. So you will see that they are showing you now MEK is doubly phosphorylated, right? Now MEK is active. MEC otherwise was not active if it was not doubly phosphorylated. Once MEC is active, then it will go and phosphorylate another protein called ERK, very, very important protein, extracellular regulatory kinase, ERK. And it turns out that MEC is a dual specificity phosphatase, okay, which means it is able to phosphorylate either a serine or threonine residue, or it can also phosphorylate a tyrosine residue. Okay, and so that's what MEC does. It will go and phosphorylate ERK on a threonine residue and a tyrosine residue. Once ERK is phosphorylated, then it is a signal for it to move into the nucleus. Okay, it will move into the nucleus and then phosphorylate a transcription factor called ALK1, for example, ELK1. And once ALK1 is phosphorylated, it will bind another protein here, for example, SRF protein, serum response factor protein. That will form a complex with ALK1, phosphorylated ALK1. And that's a signal to actually go and bind to the DNA and creates new proteins that might be required for the cell division. Okay, so that's the expression, gene expression by insulin right there. Fascinating, right? So that actually concludes chapter 12. Let me stop my recording here.